Okay, so here we are, competency 5 already out of 12. This one goes through DNA and RNA. Relatively short and sweet, so I don't want to spend too much time. This is pretty straightforward. Some of the terminology may seem a little strange, so I'll try to make it a little more clear. But for the most part, there's just a couple of processes that we really need to hammer home, and you'll find in patent chapters 4 and 7 some information on DNA, RNA. OpenStax chapter 3, not the entire, and by the way, for patent 4 and 7, OpenStax chapter 3, um, the entire chapters are not on DNA and RNA. So you'll want to take note of that. The rest of the information within those chapters is also relevant. We've covered it before, so it'll be a little bit of a review. So, but anyway, that's basically what we're covering. So, again, as always, if you have questions, please email me. Office hours Tuesdays and Thursdays. From 11 to 1:30 or 2, and then on Wednesday evenings I'm open at 5 in the Zoom room if you have questions at that time. So anyway, let's get to it. DNA, all of you have heard of that. It has to do with the genetic code, of course. We think about the nucleus of a cell containing the DNA. So what exactly is it? Well, we break it down. We see deoxyribonucleic acid. Now you know why we call it DNA. So we can break that term down even further and see deoxyribose. Okay, deoxy means we lost an oxygen somewhere. So we'll see that coming up in a second. Ribose, whenever we see that OSE on the end, we should be thinking of some sort of a sugar. So ribose happens to be what we would call like a pentose sugar, a 5-sided sugar. So anyway, OSE, indicative of a carbohydrate. And then phosphate units connecting these deoxyribose pentose sugars together. So we'll look at that in a few minutes. That forms the backbone of the DNA. Now, the center portion is going to be composed of what we call connecting nucleotides or base pairs. So we're going to see base pairs coming together, connected by hydrogen bonding. So weak bonds, remember hydrogen bonds are the weakest bonds. That comes into play in a few minutes when we're going to decode the DNA and then make copies of it and send it out of the nucleus. So hydrogen bonds, easy to break. That's good. So A binds with T, so adenine and thymine. These are the nitrogenous bases. So what that means is they contain nitrogen. So we see nucleic up here having to do with in the nucleus, by the way. I wanted to mention that as well. But anyway, adenine and thymine, and then cytosine binding to guanine. And we abbreviate these with letters. So A's always go with T's down the middle. C's always go with G's down the middle. So we'll see that coming up as well. These nucleotides are going to be organized in specific orders that are going to tell us what exactly the final product from protein synthesis that we're going to get. Is it going to be an enzyme? Is it going to be a structure? What's going on here? And it's the order of these nucleotides that determines what type of amino acid we're ultimately going to get. And then when we string amino acids together, we get a whole nother chain of codes that are going to determine what type of enzyme specifically, or what type of structure specifically we're going to get. So anyway, keep that in mind. We'll we'll go through it in a little more detail as we go. Okay. So anyway, a nucleotide, that's a pentose sugar, a phosphate group, and then that nitrogenous base, A, T, C, and G. Okay. So here we have our one nucleotide, and again, each is going to have a specific nitrogenous base of A, T, C, or G. And again, the A's are always going to connect to the T's. We have what we call a double helix formation. So basically, we have two ribbons connecting down the middle with hydrogen bonds. And again, we could see the A's and the T's, and the A could be on the other side. And if that's the case, then there's going to be a T nitrogen base on the connecting or the other side of the ribbon. We see G on this side, always going to be a C on this side. If this were a C, this would be a G. So again, you could have T and A, G and C, A and T, C and G. You can have a bunch of TAs going right down the line. You can have an order of GCs going right down the line. These do not have to alternate. They can. Again, this is a puzzle that has pretty much an infinite number of possibilities regarding combinations. So, so anyway, so keep that in mind. These do not have to be, they show one of each, but they do not have to be one of each right in a row necessarily. What does happen though is this base pairing is always going to be A and T together, or T and A together, G and C together, or C and G together. So anyway, ATCG always is that combo. So anyway, we see that 5-carbon pentose sugar with a phosphate attached, and then that nitrogenous base. Okay, that's DNA. We look at RNA, we see ribonucleic acid. So we have a straight pentose ribose sugar, not a deoxyribose. Deoxy, again, means an oxygen was removed, deoxygenated. Okay, so when we'll look at the structures of these in just a second, and you'll see that where an oxygen has been removed from the DNA, making it a deoxyribose. Okay, so anyway, this is ribonucleic acid, basically the same structure, just one oxygen lacking. So we'll compare those in a second with an illustration. Some RNA molecules are temporary copies or segments. So basically, one of the key features of RNA is that it's going to be a segment of DNA that basically it's a photocopy of a strip of one portion of DNA that's going to be decoded or translated out in the cytoplasm of the cell. So we'll look at that in more detail as well. So anyway, the idea is we're probably going to get some sort of protein synthesis going on. Proteins are going to get created. So remember, we're going to see polypeptide. We're going to see amino acids. Okay, these are the building blocks of amino acids. So these are the smallest portion of an amino acid that we're going to see here. Okay, and again, some RNA they're going to be regulatory. They're going to turn things on or shut things off. And again, some of them are going to be inhibitors potentially of gene expression. If there's a mutation noticed, RNA can help to silence or interfere with the photocopy process. So helps with viral infections. We don't want to reproduce or go through a high number of enzyme activity that's out of control, or structural construction that's out of control. We don't want that virus to be able to affect the nucleus and thus provide iffy DNA for the RNA to make photocopies of and then perpetuate the virus. So if we can stop, if we can have RNA that recognizes something's fishy when we make a photocopy, then we can maybe interfere or stop the process of continuing to make copies of viral infected DNA, so, and or proteins. So anyway, transfer RNA is just showing three. We're going to look at this in more detail. This is just kind of came out of nowhere for you. The key on this slide, don't necessarily worry about the what it is. Notice the three different representations that we see in illustrated form. You have this flat, very basic lineup here, and then we have more of a kind of like a ribbon effect going on. And then finally, what we call kind of like a 3D model or a space filler model. So you can see that ribbon, the amino acid attachment sites. An anticodon, we'll talk about what codons and anticodons are shortly. Again, really, I just threw this up here so you can see there are different ways of representing both DNA and RNA, primarily thinking about the DNA and RNA organization of all of these proteins. So anyhow, let's look at a quick kind of DNA versus RNA. Again, we see that deoxyribo, okay, and just ribo over here. So let's come down to the structure of a deoxyribose versus a ribose. We're going to focus right here. We have an OH group. Okay. Over here, we only have an H group or a hydrogen. Okay, so that's that deoxy, we've lost an oxygen over on this side. Really, that gets into some organic chemistry that you don't need to worry about, just kind of some background of why it's called deoxyribose versus ribose, lacking an oxygen. All right, so anyway, we have these base pairs lined up, and then we have one nucleotide, one base coming off of this. Okay. And then we have two other items of note that are a little bit different. We see these nucleobases, this slide calls them adenine, cytosine, and guanine. Notice both DNA and RNA are going to share those three. Okay. Now, DNA has the T we mentioned earlier, thymine, whereas RNA is going to make a photocopy where T is, but instead of thymine, it's going to photocopy something called uracil. Basically, it's the same thing. We have that functional R group, methane, coming off of there. That's basically the only difference. What that lacking functional R group does for uracil is allows for a connection to be made with adenine when we're going to make a photocopy and ultimately translate or read that photocopy. So anyhow, just remember that adenine and thymine are going to line up with DNA. With RNA, we're going to get uracil instead of thymine connecting to adenine when we make our photocopy. Now, this may not make a lot of sense at the moment, so in a few minutes, we'll go through how does DNA photocopying work. So lastly, just to cut, or not lastly, but lastly with this little section, again, nucleotides Other important roles, we see adenosine triphosphate, or ATP. Again, we have that adeno. Let's go back here, adenine. Okay, so remember, one of the base pairs, or one of the nucleobases for DNA and RNA is adenine. That rings a bell, right? Adenine, adenosine triphosphate. So there's a nucleotide, another example. And then NAD plus and FAD, which are coenzymes. They help with energy transfer. And then cyclic AMP or CAMP, also an energy molecule. So one of the key features of a nucleotide outside of DNA and RNA is thinking of cellular energy. Again, ATP, here's that adenine, the A from DNA. So basically, I don't want you to get too bogged down by some of this stuff right here. We know ATP is cellular energy. We get it by breaking a bond. And we break this P off of here, and we get ADP plus P. That breaking of the bonds, what gives us energy. We also can gain energy by consuming carbohydrates and certainly oxygen. But the point of really these next couple slides is really just to show you that we do see these nucleobases in other spots. We see adenine with ATP. We see adenine again with NAD plus. Again, this is high energy molecules. We start breaking bonds from NADH. We break that H off of there and get NAD and H. Again, breaking that bond is going to give energy. So again, I don't want you to get too bogged down by some of this intensity. I really just want you to see that adenine pops up in other spots. We see ribose popping up in other spots. And again, showing you these combined forms. They call them ATP. We have what are called lipoproteins and glycoproteins. Some of the proteins that are going to get created from protein synthesis are going to combine with lipids. Some are going to combine with sugars. So we're going to move into some of this starts to get into chapter 7 in patent. The previous DNA, RNA basics is going to be contained within chapter 4 of patent. So in chapter 3 of OpenStack. So this info is going to be found in chapter 7 of patent. So anyway, cell growth, life cycle, of course, we have cellular respiration that we'll look at. And we've touched on a little bit with ATP and the mitochondria, but we go beyond that and get into mitosis a little bit. We get into some of the basics of protein synthesis. So cells need to grow, they need to reproduce, and then that life cycle is dependent on the DNA, making an exact photocopy from the nucleus and seeing that photocopy translated and a new amino acid group being put together to make a polypeptide, being put together to make a protein, whether it's an enzyme, whether it's a structure. So basically, we're ensuring that the genetic code directed from cell to future cells is going to be identical. Unless, again, we have a viral infection or an autoimmune disease, then something completely that we need to stop the progression somehow. So, and cancer too, of course, is this cell replication gone haywire. So anyway, it's the primary anabolic or building pathway in cells. We're taking small bits, amino acids, making polypeptides and making proteins. So even smaller than those amino acids, we can take these base pairs, break them apart, get a little map of what we want to create, and translate that map or that code, and go from there. So anyway, DNA, again, a double helix composed of those nucleotides that has the genetic code. And one gene is going to be a segment of DNA that's going to contain approximately 1,000 pairs of nucleotides. So one gene contains roughly 1,000 base pairs. So just to give you an idea of what a gene is, very tiny. But when we break it down, we see around 1,000 of these A, T, C's, and G's combined together in different patterns to create some sort of structure or chemical. Watson-Crick were the first group. Some of you have done your case study 5, so you'll learn a little bit more about those two. So what does a DNA molecule do? Why do we have genes? Again, each DNA molecule is made up of millions of different genes. Each gene is made up of thousands or around 1,000 of these base pairs. Okay, so we're going to make photocopies in the form of RNA transcripts. Okay. So we're going to transcribe, and some of the RNA is non-coding, so it's going to be supportive. We'll look at some of those specific types of non-coding RNA. And then we'll look at the specific coding types of RNA that are going to dictate protein synthesis, which again, protein synthesis occurring in with the ribosomes and the rough ER are going to ultimately create some sort of structural proteins or some sort of enzymatic proteins. So the coding RNA is called messenger RNA. These are what makes the photocopy in the nucleus. Okay, so we transcribe with messenger RNA or make a photocopy inside of the nucleus, and then outside of the nucleus we have non-coding RNA. These are the helpers. They help read or translate in the cytoplasm what the map says. So these are ribosomal and transfer RNA. Okay, so transcription occurs in the nucleus, messenger RNA making a photocopy. Okay, so that's what's going on here, transcription. So again, we see the DNA unzips, and we have these little RNA nucleotides kind of floating around inside the nucleoplasm or inside of the nucleus. As soon as a DNA molecule unzips, we're going to see this side get exposed, this side get exposed. So one side of it is going to start getting the matches. So we see the C's coming with the G's coming in with the C's. And remember, there are no tRNA nucleotides. We have U's, uracil, so those come in with the A's come in with the T's, and so on. So we get this little strip that got unzipped here, and then we're going to connect some RNA nucleotides, which then form a strand. And that strand is going to come off the copy machine or come off the line, and we're going to then have the matching opposite code that can be translated in the cytoplasm with the help of the ribosome. So we can transcribe or make a photocopy. This RNA strand now has the opposite base pair, so it's going, or opposite nucleotide. So we're going to go out and make a new structure that's going to contain the map of what ultimately becomes a protein or a polypeptide. We see the words intron and exon when RNA does its splicing. So some of this information that's in here is going to be irrelevant. Introns are going to get pulled and put in the trash. Exons are what get expressed. So when you see exons, that's what ultimately gets expressed in the mature messenger RNA. So there's another step that goes on. It talks about it, was it in this one? I think it's in another slide. It talks a little bit about, oh, right here, editing the RNA transcript. So we have that DNA chunk, we get the RNA, messenger RNA coming in and getting a preliminary transcription of the opposite side of these nucleotides. And then some of these chunks, these orange chunks in this particular illustration, are going to be considered introns or trash. So we're going to pull those out and then splice together all of these exons. And then we're going to pinch off the end. It talks about G nucleotide and poly A. That just means we've got a couple little caps that we can put on the end. And then at this point, we have our strip of messenger RNA that is going to be able to be translated out here in the cytoplasm. Okay, so again, introns are chunks that get pulled out. And we get editing going on, and then we can transport the final product. And again, viruses can come in and they can affect the recognition of what's an intron versus an exon. This code may be completely altered by a virus, so the photocopy itself is going to be different than what the nucleus would normally be pumping out. And again, confusion with introns and exons. Then we get a recipe that is delivered to the cytoplasm, to the ribosomes and the rough ER, and that ends up getting created. And now we have a cake with a whole bunch of baking soda instead of a whole bunch of flour, for instance. And so that's not going to be a very solid or functional protein. There are going to be chemical issues potentially. So anyhow, that's what a virus is going to be doing. So next chapter, next competency is on the immune system. So we'll talk a little bit more about how viruses can affect the editing process of messenger RNA. So anyway, we're going to leave the nucleus, go into the cytoplasm, and translate that messenger RNA photocopy with the help of ribosomes and transfer RNA molecules. These are going to be bringing some of the amino acids in that are going to help form the protein chain. So as we're making a photocopy, the transfer RNA helps with transferring the product and getting it ultimately placed into the next phase of construction. And again, rough ER is going to help with this. Golgi apparatus gets involved with some of this as well. Okay, so as these amino acids get brought into place, that's when we can form peptide bonds. That's where we ultimately form a polypeptide chain. So one thing that happens when we translate these genes is we're going to see that those nucleotides, those A, T, C's, and G's are going to be taken in groups of 3. And so keep that in mind as we go as well. So anyway, this is a little bit fuzzy, but I think you can see what's going on. It's in your patent set. I think it's figure 7-15 maybe. But anyhow, we see the transcribing going on in the nucleus. Messenger RNA comes out, and we get these ribosomal subunits that are going to basically clamp and connect together And we're going to start zipping things up and basically waiting for transfer RNA to bring in the right, what we call codon. So an amino acid is going to be created, in essence, by matching up these organizations of three uh, nucleotides forming a codon. So I'll show you how they do that, and or how we do that in just a second. So you can see the transfer RNA is bringing in a trio that's going to match up exactly with whatever RNA uh, ribbon or blueprint recipe that has been delivered from the nucleus. Okay. So codon is a group of three. We'll look at an example of that. So as these come together, we're going to get, as these transfer RNAs come in, they're going to connect with the appropriate codon, and then we can start stringing these amino acids together and form polypeptides. So we can see the polypeptide bonds forming here. Okay, so this guy is going to come in and attach with this group. There's a G, an A, and a C. Yep, so sure enough, here's the next trio. You can see this is a C, an A, and a C. So this would have to connect. The C would have to connect with a G. The A connects with a U, and the C connects with the G. So that's going to be the next codon, and that's going to be a specific amino acid. And that specific amino acid is going to be shown right here by this codon. So CAC, that order, first base, second base, third base, that's going to be the specific code or codon for a specific amino acid. And there are 20 different amino acids. Okay, so this particular CAC is a specific amino acid. Okay, so that's going to come in, and then you can see it forms a connection with the ones that came before it. And then what we get is the transfer RNA leaves after it drops off. You can see it's bringing in an amino acid, allowing it to attach and form a polypeptide bond, and then we see that transfer RNA leaving. So, hi, Crystal. You can stay here if you want. I'm just about done. I'm just recording a little DNA RNA competency 5 video. It'll just be another minute or two.
Okay, no problem.
Okay, thanks. Okay, so one example of this codon that we're going to look at. So what amino acid is this? What amino acid is this? There are, again, 20 possibilities. So what we do, we're going to take, for example, we'll take C, A, and G. They give us C, A, and C down there. Oh, and by the way, this could be a stop codon. So we may want to stop the process of building this polypeptide chain. So this could be a stop code. So what they've developed to figure out the genetic code is, remember, you have that first base, and then that second base, and then that third base. So we're going to do C, G, and A. So when you have the trio C, G, and A, or the codon CGA, you start at the top, C. So this is the box we're going to use. And by the way, all of these are the 20 different amino acids. So what does CGA code for? What specific amino acid? If I had CGA floating around, what specific amino acid will be brought in? So C, so it could be any one of these. The second base pair we call G, so it's going to be here. So that goes to here. Now notice this has 2, this has 2, this one has 1, so we could pretty much stop there. Anything that's CG is always going to be arginine or arginine. But for third base codon was A down here, so then we go up and end up in the same spot. So CGA puts you here in arginine or arginine. If we go back here and see CAC, we're going to see C, A, and then C. So histine or histidine. Okay. Oh, wait, that's CAC. Did I do CAG? I did. Yeah, CAC, histine. Okay, so if we did have one that was UGA, for instance, or UAG, that would be a stop codon. So, and again, how do we know what this is going to be? That comes from this opposite, the opposite nucleotide or base pair. Okay. And again, all of this occurs afterwards in the rough ER and the Golgi apparatus. And the last section we did in general A&P, so I'm not going to go through mitosis. Just remember, interphase is the bulk of the lifespan of a cell. And then we have the mitotic phase, or the cell division phase. PMAT, we called it, pro, meta, ana, and telophase. Remember though, the bulk of the lifespan is spent in interphase, the G1 phase, the S phase, where we have cell division taking place, and then the G2 phase, or cell replication, I should say, DNA replication. Cell division happens up here. This is the DNA replication phase. So all of that DNA replication that we just saw going on with protein synthesis, we can also get the replication aspects that are contained within the nucleus of the cell that ultimately lead to cell division. So not only protein synthesis regarding DNA, but also cell division regarding DNA. So if you have questions, email me or join on Tuesdays, Thursdays from 11 to 1:30, or Wednesdays at 5.